My name is Mike Pishvaian. I'm an associate professor in the Department of Oncology at Johns Hopkins University School of Medicine. One of the most exciting areas of study in pancreatic cancer are the pancreatic cancer patients whose tumors harbor mutations in the DNA damage response and repair pathway. And this accounts for 17 to 25% of pancreatic cancer patients. So it's not a small subgroup. And more importantly, it's a subgroup of patients for whom there are new therapies being developed that could make a significant impact in their outcomes. So what is the DNA damage response and repair, or DDR pathway? Well, it's a very well conserved, evolutionarily conserved pathway of multiple, upwards of 80 enzymes that have one goal, which is to maintain the integrity of the DNA code as cells divide. And the DNA undergoes multiple insults as cells divide, environmentally, from UV light and chemicals that we ingest, and then certainly from things that we're trying to do, like chemotherapy or radiation therapy, where we're trying to damage the DNA of cancer cells. And cells have a very elaborate system for repairing that damage. So even either single-stranded breaks that get repaired from the nucleotide excision repair pathway or the base excision repair pathway, to double-stranded breaks that get repaired by homologous recombination or non-homologous end joining. These are systems that are well put in place to try and allow the cell to maintain the normal DNA integrity. This is critical for our normal cells, but also especially for cancer cells, because they're dividing really rapidly. And if they divide rapidly but undergo excessive DNA damage, then even the cancer cells will eventually essentially implode. The degree of DNA damage will not be sustainable. And so the cancer cells also need to maintain these DNA damage response and repair pathways optimally. So we also have several ways to target this pathway, and we hope that they are Achilles heels of the DNA damage response and repair pathway. So for example, we know that PARP inhibitors work both in the single-stranded break repair setting, but they also work in preventing homologous recombination by latching onto the DNA strand and preventing the replication fork from moving forward and causing replication fork arrest. There are inhibitors of drugs that sense the DNA damage, inhibitors of enzymes that sense the DNA damage. So ATM and ATR are critical enzymes that will sense DNA damage, and blocking those enzymes can prevent the DDR response from going forward. The DDR response takes time, so the cell cycle is usually arrested by the cell to allow time for the DNA damage to be repaired. So if we prevent the cell cycle arrest with CHEK1 inhibitors, with WEE1 inhibitors, and drugs that are targeting that pathway, then we force the cell to go through cell cycling more rapidly than it's able to. And so as a result, the DNA damage is not able to be repaired. And then finally, we now have newer drugs that are targeting the sort of error-prone, non-homologous end joining pathway. So when cells have the inability to repair damage through the more well-conserved homologous recombination pathway, they then will revert to the non-homologous end joining pathway. And there's a critical enzyme in that pathway, DNA-PK, that is necessary for that pathway to move forward. And we now have DNA-PK inhibitors, which are just now entering clinical trials, and we're hopeful they will also help help be part of the armamentarium for treating patients with cancers. But pancreatic cancer patients harbor about 17 to 25% of the time either germline or somatic mutations in this DDR pathway. The most common mutation actually is an ATM. About 2% of the time it's germline, an additional 4% of the time it's somatic only, meaning in the tumor only, but it is the most common mutation in the DDR pathway. The second most common mutations are in BRCA1 and BRCA2, the ones that are probably known the most about. And then there's a whole slew of additional genes in the DDR pathway that we see mutated in pancreatic cancer. And in many ways, we assume, although we don't know yet for sure, that mutations in those critical enzymes and the genes in those critical enzymes similarly confer responsiveness to different therapies like PARP inhibitors, like platinum-based therapy. But over time, we're going to have to figure that out.